Welcome to the 22nd seminar of this series from the International Tree Mortality Network. I'm really happy that today we get to welcome Lalasia Bialic-Murphy from the Growth Lab in ETH Zurich, where she's a lead scientist. Lalasia works on linking population and community ecology, and really excited to hear the work that she's been doing in the last few years about tree longevity. Just a few words about how we run the seminars in case you haven't been here before. So Lalasia will present her work, and your questions are really welcomed. We'll have a good portion of time after she's presented it for a question and answer. If you please just put your questions into the Q&A box at the bottom of the screen. You can do this at any time during the talk or after the talk, and we will collate those and then try and ask as many of them as possible within the time that we have. So without any further ado, Lalasia, I will hand over to you. Looking forward.
So thank you very much for being here. I really enjoyed these seminar series, and I'm honored to be able to present to you some of the work that I've been thinking about for the last several years. So this project is part of a huge collaboration with international ecologists from around the world. And I'm going to be focusing primarily on a project we're calling the Pace of Life for Forest Trees. To give you a bit of a context of who I am, I'm, as Tom mentioned, I'm Lalasia Bialic-Murphy, and I'm a lead scientist within the Crowther Lab. This is my amazing team in the fundamental ecology group. So we're nested within this larger group that studies a lot of different ecological scales, as well as several different groups that work on the intersection between applied restoration and conservation management, as well as policy, and how we can use academic research to help inform these larger motivations or these larger projects in the applied realm. Within the fundamental ecology group, I don't know why, within the fundamental ecology group, our work we study the mechanisms, processes, and principles that give rise to biologic diversity and ecosystem function. And this is really scaling and thinking about species level responses with other organisms, as well as the environment, so the abiotic environment, all the way up to community assembly and coexistence to ecosystem level processes. So it's this eloquent dance between the biotic and the abiotic environment and how that shifts over time in space. I like to, in setting this work, kind of scale back to what we can say really confidently within ecology. And one of the things that I think are pretty strong emergent patterns that we can say is that as you go across forest systems, so from the tropics up into the more extreme environments, we see this strong productivity gradient with really high productivity in the tropical regions and then diminishing productivity in these more extreme environments. And what I mean by extreme is more the abiotic processes that are regulating metabolic processes for vegetation, so temperature, soil, and precipitation. And at the same time, there's a lot of really great work that has shown that productivity is really strongly related to diversity patterns across forest systems. So again, really high diversity in the tropics and diminishing diversity as you get into these more extreme environments. And this has led to a lot of amazing work over the last several decades in looking at this relationship from both an evolutionary lens, so thinking about productivity driving diversification of systems, as well as from an ecosystem functioning perspective, so more diverse systems being more productive. And the idea behind that is that within a static pool of resources, species with different morphologic and physiologic traits have a higher ability to access and utilize more of those static resources, and thus being more productive. So again, we see this really kind of strong relationship between the diversity and productivity from both an evolutionary and an ecosystem functioning perspective. I don't know why this is. Within the Crowther Lab, one of the things that we've studied over the last several decades, and this is not my work, this is some work by Ledong Mo et al., has shown that when we look across forest systems, that humans have had a huge impact on the standing biomass of carbon across latitudinal gradients. And this has garnered a lot of attention in thinking about how do we restore and protect the remaining intact systems as well as the degraded systems of forest as one of many climate change mitigation strategies. And there's also been a lot of research in thinking about the second part of this feedback. So how does the last centuries or decades of climate shifts, as well as future projections, gonna feed back into influencing the dynamics of these forest systems? So the productivity of carbon as well as nutrients and water fluxes. So we start scaling and thinking about this interplay and feedback between uncertainties in scaling, the speed of carbon sequestration, following habitat degradation and how that may vary across latitudinal gradients, as well as starting to model shifts in carbon turnover rates. This is centrally underpinned by demographic processes. So the relationship between how fast trees and dominant trees within communities and across ecosystems are relating to how fast they're sequestering carbon in plant growth and how that relates to mortality and longevity of trees. And so this has been really nicely shown by several different studies and thinking about several different hypotheses of the relationship. So for example, in hypothesis 2, you could think about a climate variable that's stimulating plant growth, so accelerating carbon sequestration. And in the context of that not being jointly trading off with longevity, you could see that climate variable would be speeding up the system, but having similar residence time. Conversely, in hypothesis 3, you could be thinking about a system or the environment speeding up carbon sequestration, but if that's strongly trading off with reduced survival and lower life expectancy, you're talking about entire systems just speeding up. In thinking about either projecting the time of recovery across latitudinal gradients and or the effects of climate change, there's several underpinning assumptions that are really difficult when scaling. And this has been really nicely shown by large vegetation models, so different types of vegetation models represented in this figure, where Tom et al. Showed that the parameterization of the growth dynamics and how that relates to mortality and longevity can lead to highly different turnover rates across systems across these latitudinal gradients. So varying just by looking at the color coding, which is representing turnover rates of carbon throughout systems. So to start getting a more concrete understanding of tree growth, longevity, and stature relationships across large-scale resource gradients, I've been collaborating with a huge team of international partners, and this has been an effort that's been led by the Tree Mort group or Tree Mort effort, where they've been collating dynamical data, so survival and growth data for trees across the existing forest inventories across the world. So that's including forest inventories from Canada, from the United States, as well as Europe, as well. And then in the Southern Hemisphere, a lot of effort by the Forest Plots Network, and that's composed of a whole bunch of different plots from around the world and people collecting dynamical data. So this is a huge effort, and I'm only a small part of it. So using these long-term dynamical data, the effort that I'll be talking about today is really mapping out quantitative metrics in the relationship between the speed of carbon sequestration and plant growth. And from a demography's perspective, we can quantify this by looking at the number of years that it takes a particular species to reach calibrated size thresholds. So for example, we could be looking at the number of years a tree goes from that it takes for a tree to grow from 10 centimeters to 20 centimeters. And that would give us a metric of comparison in the rate of carbon sequestration among taxa. Similarly, we can quantify the number of years it takes trees to reach their maximal size threshold. And this is a metric that captures the growth dynamics over a species life cycle. And that'll be highly variable among taxa because species reach very different sizes and maximal statures. So these are two metrics that throughout my presentation, when I'm talking about growth and the speed of growth, these are the metrics I'm talking about. The other metric that I think is really important in thinking about scaling from species level responses to communities and ecosystems is life expectancy. So this is really related to carbon residence time within a system. And using cross-sectional forest inventory data, we can quantify species-specific life expectancies. So this is based on hundreds and thousands of observations of individual trees that allow us to quantify the mean life expectancy of trees and how that varies among species within and among environments. We can also quantify the maximal lifespan. So the maximal number of years the luckiest individuals of that species will live. To give you a sense of how we quantify these life history traits, we used a size-dependent integral projection model. So it's a species-specific model that's represented in this equation in the upper equation here. And in the heart of that equation is the PYX function, which is just simply a series of regression models. So it can be decomposed into the size-based probability of surviving among thousands of individuals and the size-based probability of growing. And then using those regression coefficients, we can integrate from the upper to the lower size bound. So this would be the maximal size of a species, and the lower size bound for all of the comparisons in this study is 10 centimeters. Through that integration, we can get a 3D kind of surface probability of a species movement. So from size at time t to t+1, represented on the two x-axes, and then a probability of movement. So in the lighter green colors, there's very low probability. So, and then in the higher heat tones, that's representing a really high probability. And then by incorporating mortality as an absorbing state and using age from stage analyses, we can convert a probability of event to a time of event. And I think this is a really eloquent way to convert what we know is the best predictor of mortality and growth for trees, which is size, to a time that's more related to these higher order ecological processes. So using this approach, we use the dynamical data across the Americas. So we're talking about a latitudinal gradient from Brazil, southern Brazil, to up to the northern part of Canada. And these dynamic data included about 3.2 million observations and incorporated quantifying life history traits for about 1,000 species, a little over 1,000 species. Because multiple species span more than one of these larger tiles, which are 250,000 kilometers, so relatively large scale. When species represent or span multiple grid cells, we can quantify within species variation and life expectancy across this latitudinal gradient. So doing this, we developed several different expectations, and this is really the first expectation is driven by demography and evolutionary theory. And it's been kind of coined over and over with slightly different viewpoints. The earliest kind of expectation would be RK selection, so qualitative kind of categorization of species from the fast to the slow growing species and how that relates to the life expectancy. Then we incorporate RK or the Grimes triangle, and then more recently it's been referred to as the fast-slow continuum. But based on any of those theories, the major expectation is that really fast-growing species should have shorter lifespans and that slow-growing species should have longer lifespans. So species should fall along this continuum. And in the green on the upper left, we would expect that species wouldn't occupy this space because of ubiquitous assumptions of resource limitations. So if species are in this kind of continuum or this space, we would kind of coin them the Darwinian demons, the ones that are doing everything well and just can outcompete every other taxa within the environment. And then at the other end in this red color, we would expect those species to be maladapted over evolutionary time, so outcompeted by other taxa. And this general expectation has been shown, kind of the diversification of species along this continuum has been shown by looking at selection potential for morphologic and physiologic traits that allow species to maintain really fast growth versus survival. And it's shown that species that are short-lived tend to have really high selection potential for growth, so accelerated growth. And conversely, longer-lived species have this high selection potential for higher survival. So you see this same kind of continuum and thinking about it from a selection potential viewpoint on optimizing a species fitness. Kind of scaling out from this really simplistic viewpoint of the continuum, the fast-slow continuum, and how that may map out over biogeographic gradients, we would expect that as species are diversifying and or just occupying different environments, that there should be really strong trade-offs between these traits. So when I'm talking about trade-offs, we're talking about the trade-off between growth, longevity, and stature. So for example, if you're going from cold environments to warmer environments, you would expect that species would be stimulated as far as plant growth, but that would come at the cost of lower life expectancy. And then scaling even more to macro scale. And so this is looking at community dynamics of life history strategies. One of the expectations is that as you go across both productivity and resource gradients, that we should see really strong signatures of the volume of strategies that can persist within a particular environment. So this really goes along with the same expectation in macroecology, where in these more extreme boreal environments, you should have constraints on the range of life history strategies that can persist. And then as you go into more productive systems with higher temperatures or intermediate temperatures, that you should see more selection for differentiation and competition and niche and kind of an increase in the volume of strategies within those environments. So now kind of going through some of the results that we find, I first just mapped out and compared patterns in the average life expectancy for all of the species within the tropics versus the extratropics. And we find that on average, the life expectancy is quite a bit higher, so not double, but close to that in the extratropics. And this really mirrors the information of the core hotspots of the longest living trees, so the sentinel oldest living trees, which are commonly found in the temperate conifer forests and in the boreal regions. Moving on and thinking about the relationship between growth, which is represented in panel B here, where we have the growth of trees going from 10 to 20 centimeters, and the life expectancy. So this is the years in growth and the years in life expectancy. And one of the things that really motivated a lot of the subsequent results that I'll show you is this basic scatter diagram, where you can see that there's really not a strong alignment in what you would expect based on the fast-slow continuum, where species that have really similar growth strategies, so take the same number of years to grow to 20 centimeters, have this wide range of life expectancies. And you can also see visual patterns in the variation among different kind of biome types, so the tropics versus the boreal regions. So in order to help simplify this variation in demographic strategies, what we did is first just mapping out a principal component analysis and comparing the growth to 20 centimeters and the growth to 20th, or the growth to the 20th centimeters and the growth to the 70th quantile of the species size range, the life expectancy and the maximal size. And you can look at the variance covariance among the traits across these PC axes. One of the things that I really want to point out is not necessarily the weighting. So the weighting is important in some regards. You have growth really being heavily weighted on PC1, size PC2, and life expectancy on PC3. But the relationship, the covariance between the traits flips as you go from one PC axis to the next. So this is a strong indication that there's more complex dimensionality in the relationship between these life history traits. So using the first 3 PC axes for all of the taxa, we then did a clustering analysis, which allows us to simplify thousands of species into core demographic functional forms. So here are 2 of the kind of visual results of that clustering analysis. When you look at the first 3 PC axes, species aggregate into 4 different demographic functional types. And I'm going to go through this a little bit slowly because I think PC analyses can be a little bit unintuitive to look at first glance. So PC1, it's heavily weighted by growth, from really fast growth to slow growth. And then PC2 is from small size to large sizes. And you see that the really fast growing species are constrained to intermediate sizes, maximal sizes. And then as you go out into these conservative, slow growing species, you see separation into 3 different functional forms. Then moving to PC2 and PC3, another of the kind of main results that we see is that this really slow growing conservative species are the longest living taxa. So these give you like the functional forms that are relating to that relationship between carbon sequestration and carbon retention, and showing that the slower growing species can separate out into very different life expectancies and residence time of carbon. We see the same kind of relationship in the PC axes when we look at just the species in the tropics versus the extratropics. But one of the main results is that when you then use those PC analyses to cluster them, the extratropics converge into 2 to 3 different clusters of more conservative taxa, and that the tropical species maintain these 4 different functional groups of species. So now moving on to thinking about environmental gradients and the life history traits. We set up a simple Bayesian model, which allowed us to control for evolutionary history among species, and then mapping out the influence of the abiotic environments in shaping these life history traits. And then more importantly, how these traits covary across environmental gradients. So using this approach, this is one of the main results in the coefficients. So this is the coefficients of the Bayesian model. 0 represents really no signature of the soil precipitation and temperature variables, and confidence intervals that deviate from that 0 line represent a strong signal. And what we see here is that just visually is that temperature is really having the most pronounced effect on both of these growth strategies, so growth to 20 and growth to 70th, as well as maximal size and life expectancy, with less variation explained by temperature, by soil and precipitation. And I think this really closely mirrors a lot of the global studies that look at temperature really regulating macro scale patterns. And you can see visually the same kind of coefficients represented here in just these scatter diagrams of looking at temperature on the x-axis, and then you have your growth strategy, your maximal sizes, and your life expectancy, with slower growing species in the boreal and then faster growing species in the tropics. Also notice the variation, which I'll also be talking about in a minute. But this gives you a sense of the general patterns across biogeographic gradients. And I would argue maybe more importantly than these signatures of the pace of life of trees across these biogeographic gradients is the covariance or the coordination between life history traits. So when you look at growth to 20th centimeters and growth to 70th, there's a really strong covariance, so a strong relationship between these variables. But then when you start looking at life expectancy and how that relates to growth and size, maximal size, there's relatively a low signal, or a low kind of a weak coordination between these traits. So this suggests fundamentally different relationships between the speed of carbon sequestration in plant growth and carbon retention in plant longevity when you're scaling across these biogeographic gradients of temperature. Okay, so now moving on to looking at and mapping out the trait volume of strategies across latitudinal gradients. How we did this is across each one of these tiles, we're representing the life history traits for all species in that environment. So their growth strategies and their life expectancy and their maximal sizes. And then using those variables and the PC analyses of the trait correlations between those variables for all species within each grid cell, we mapped out the convex hull volume. So it's the volume of strategies represented by that grid cell. So using this convex hull approach, that's represented what I'm calling the demographic trait diversity. So it's the volume of demographic strategies. I first wanted to do a sanity check, really, to look at the relationship between species richness and demographic trait diversity. And what we find is that there's a really strong positive relationship between richness and demographic diversity. And so it's inferring that with the species that we've been able to capture with this study, that we're really seeing biogeographic patterns that are similar to what we know is happening in ecology, which is high richness to low richness as you go from tropics to boreal. But one of the cool things is that we can look at the volume of strategies and condense that variation into one trait volume versus the dynamics of hundreds of thousands of species. We also see that similar pattern in the volume of strategies across a temperature gradient. So now moving on to the main results of that I think are maybe more interesting. If you look at the panel on the left, we mapped out and tested the expectation that productivity should drive diversification and niche differentiation. So the volume of strategies that we see across biogeographic gradients. And what we see is that as you go from the boreal regions, which is represented in this reddish color, the orangish color, to more productive systems, you have higher diversity of demographic strategies. So this is really consistent from this lens of evolutionary ecology of productivity driving diversification of systems. And then similarly, if we just flip those axes and look at diversity predicting productivity, I used a basic mixed model. And after controlling for temperature, or after including temperature, we see a strong significant effect of demographic diversity predicting productivity. So this really mirrors a lot of work that's showing species richness predicting productivity, but it gives us a more direct link between these demographic strategies are really regulating carbon dynamics. So the growth and the longevity of trees and how that relates to the productivity of systems. So this macro scale study really doesn't confirm the causality of this relationship, but it just establishes that there is a signature of the demographic diversity of systems and productivity across these large-scale latitudinal gradients. We can see this consistent kind of pattern when we look at species within just the tropics versus the extratropics. So this general pattern isn't just driven by large-scale resource gradients, but within the tropics versus extratropics, we see this same positive relationship between diversity and productivity. So I've rambled on a bit here, and I'm just going to distill the key points of what I think are really important in scaling. So what we've learned from this work in scaling. And the first point is that we can map out across thousands of species the dominant functional forms of taxa into three different clusters in the boreal regions, depending on the clustering algorithm we use, to 4 different functional forms in the tropics. And these functional forms are really related to the core strategies that are regulating the dynamics of those systems. So it simplifies hundreds of species and their dynamics into these functional forms of taxa. The second key take home is that this relationship between the traits of thinking of the relationship between how fast carbon is being sequestered in plant growth and how that relates to longevity and mortality of trees is fundamentally different across latitudinal gradients. And so we really need to incorporate and consider that signature and the strength of that interaction across these gradients in order to narrow some of the uncertainty in larger scale models. I forgot to turn off this video every time. The third kind of scaling implications is that we find predictable variation in demographic trait diversity across systems, and this has important insights that build on well-established diversity productivity relationships, but it gives us a more direct link across these scales. And so with that, those are the main results that I want to share with you today. I also want to acknowledge all of the amazing collaborators that have contributed to this work. So Adriana, Tom, and Oliver really introduced me to this huge community of ecologists that are collating dynamical data and thinking about the same kind of processes that I am. And so it gave me the opportunity to be introduced to this group and to collaborate with them, as well as Tom, Robert, and Johan from our lab at ETH, so Crother's lab at ETH, that have contributed to this work in various ways, as well as all of the members of the forest plots and the tree mort teams that have contributed intellectually to the work, as well as been willing to share all of their amazing data for this project. So with that, I want to thank you for your time, and I'm happy to take questions.
Fantastic. Thank you, Lalasia. Really nice. And it's a really, really cool topic. We haven't really seen anything like this in our seminar series so far. So really nice. And I can say for myself that as someone who spends their time trying to model these ecosystems, it's really, really exciting to have some different takes on these functional types held together by all of this data. So now we have some time for questions. If people watching, you have questions you'd like to ask Lalasia, please just put them in the Q&A. While you're thinking about that, I'd like to start off a little bit. And so you showed Lalasia these really interesting linkages or non-linkages between size and growth and longevity. Where does reproduction fit into this picture?
Well, first, there's a lot of really cool work from tropical tree forest plots predominantly, but also some from the temperate zone that show that that is a third axis of variation, so that the growth and longevity, or survival, growth, and reproduction all trade off. So energetically, they're trading off between the allocation of those resources. And that when you're thinking about scaling from the growth strategy over a species life cycle, longevity, what you would want to measure is the net reproductive rate of a taxa over their life cycle. So how much they're investing in reproduction over all of the years, right? And so we could incorporate that into these models, but we just don't have the type of demographic data that we need right now to incorporate that into these dynamical models and look at net reproductive rate of species. But it could help explain where some of the species are following in that axis of the relationship between growth and longevity, as far as that they're allocating towards reproduction, and that's why they have that decoupling. And that's been shown using demographic data sets for herbaceous species, but for trees, there's only a few studies that have looked at that, and it's at the local scale because you have more data for that.
Do you think there's a perspective to really answer that at the large scale, or always going to be limited by data too strongly to be able to address it empirically?
I think that there are ways of doing it. There's some massive assumptions, but I think that we could start getting a better understanding of the incursion rate of trees getting tagged into the 10 centimeters and make some kind of calibration of how many years it's taken those trees to enter that stage class as a reproduction on an annual basis. That could be used, I think, with the data we have. The thing that is the most challenging is that trees start reproducing at very different sizes. And so we're just starting to get a sense at what size plants start reproducing and how that varies. And with that information, I think we could start modeling it with some assumptions, but right now we just don't have those kind of data that I'm aware of. If you have it, anybody on here, let's collaborate, but I haven't found it.
Cool. Okay, I'm going to pass over to a question from Giuliano Cabral, who says, so if temperature emerges as a key predictor on some of these traits, has the relationship also been tested across elevational gradients as well as latitudinal ones? Is that something you've looked at?
We're starting to look at that. I don't have a full picture of that now, but you would think that it would relate. I mean, it kind of is related in the temperature because you have elevation influencing local temperature and precipitation in soil, but we haven't looked at the kind of independent effect of elevation. But yeah, that would be a cool thing to do.
And so there's a question linking to that in a way. So linking in not only temperature, but also water availability. So one question is, you mentioned the global productivity diversity relationship. Isn't it simply a consequence of the high ecosystem water availability and optimum temperatures for growth that cause the high diversity? How does this all map together?
Yeah, I think that this is a challenge more generally because we think about like we've looked at temperature within temperature, precipitation, soil. We included all the kind of main layers within each of those categories, and then it did a PC analysis and used the values of that PC1 to model the signature. The reason we did that is because they're highly correlated, all those traits. And so if you get instability in your models, so it's really that you can't separate them out in some regards. In thinking about the kind of interplay between those core groups, so temperature and precipitation, of course, for physiologic processes, they're interrelated. At the global scale, when you look at precipitation, temperature, and soil variables, temperature's really sticking out. And I think that makes somewhat intuitive sense when you're thinking about metabolic processes of photosynthesis. So what's activating photosynthesis? And maybe mortality is more related to precipitation, but we don't see that signature at a global scale. That doesn't mean, of course, they're interacting, and temperatures, the importance of temperature, the limitation of temperature on physiology is going to be related to precipitation. But I think that because these variables are so correlated, it's going to be really hard to separate them out at a global scale. But it's something that we've been playing around with quite a bit in modeling. We're now kind of extending these models to incorporate within species as well as climate fluctuations of, do we see independent responses to temperature versus soil or an interaction between them? And then we're doing that to think about future climate scenarios. But yeah, it's a big challenge.
Do you think the challenge is also exacerbated by the samples that are available? For instance, if you had more plots data available in dry forests in the tropics, do you think that might change a bit the picture?
Yeah, I mean, of course, I think that what we're starting to be able to get with these people with these networks and sharing, particularly in the Southern Hemisphere, we're starting to be able to get somewhat of a sense of it. But of course, the Northern Hemisphere is way more studied and has more detail, so mapped out across a wider range. And so to get that level of variation within the tropical systems, we need more and more data to really separate that out. And also it's something that, here we're talking about pretty large scale gradients, but I think that we could look at within region too, and look at the effects of temperatures and precipitation signatures within those bounds. And so I think that could be another interesting study to separate large scale gradients versus local scale gradients. We kind of started looking at that with just looking at dynamics in the tropics versus extratropics. And we see that same pattern of higher diversity and higher productivity in the tropics. And so it's not just a signature of macro scale, but within those regions. But to get even at a more local scale would also be really interesting.
Oh, that sounds really cool. Okay. So a question, I think this question is from Bernhard, actually. And asking, did you look at all, or were you able to separate between early versus late successional species, or drought avoiding versus drought tolerating species when you did the analysis? I suppose we could expand that. Did you cross-reference with any other traits, what beyond the demographic ones?
I really want to do that. Of course, there should conceptually, from an evolutionary perspective, you would expect that demographic traits and fitness should be strongly related to morphologic and physiologic traits. So I think that that's a cool extension of this work, and looking at what we're theoretically expecting, selection for morphologic, physiologic traits that allow species to maintain high growth or high longevity, for example. So there should be a direct relationship. We didn't do that here, but it's a natural extension of it. As far as early and later successional species, there has been some very cool work in the tropics that have shown that. So looking at successional recovery of systems. Here, we chose to use larger grid cells, 250-kilometer grid cells, because we're wanting to look at macro patterns. And also there's some complexity in the type of modeling we're doing. So a lot of times when we're looking at broad scale patterns, you're looking at average community level mortality rates or average growth dynamics. But for models looking at, you have to have survival and growth variation across a species size range. So you really can't calculate longevity for a system that's been heavily disturbed and just after recovery, because you only have individuals of the smaller size class. So you have to have some variation in the size distribution of the taxa to get a meaningful life expectancy for those species. But one way to kind of look at that is kind of from the back angle of looking at time since disturbance and which species are coming in. Do you see a pattern of the longer living species versus shorter living species? And that could be cool to do.
So linking to that point about disturbance, then, do you see management as a big challenge here? Does management not really matter so long as you have a range of tree species, tree individuals across the size range? Or is it something that could change the relationships that you see?
I think that there's one of the earlier seminars that I watched was talking about within species variation and longevity, and they were using tree ring data to look at that. And I think that conceptually makes a lot of sense, because you're talking about a lot of variables like light gaps and things like that drive within species variation in their demographics. Because we're looking at between species, I would expect that the variation within taxa is going to be narrower than the variation between taxa. So because it's a species level average and we're using thousands of observations across a whole bunch of plots to parameterize an average for that taxa, we're getting it kind of across successional stages. But there's most definitely probably within species variation. It gets a little bit hard for me to think about, and when you're talking about a tree that lives 4,000 years old, that is living, an individual tree is living through succession. And so there is some complexity of time of a cohort that's born into a disturbed, highly disturbed environment is living through the time period where you have the old growth forest coming in. So yes, it's going to influence their life expectancy, but it's also the time scales that would be hard to incorporate into these larger models, I guess.
Oh, okay. Thank you. So I have two more questions here. And this question is from Omar. Don't know if Omar has a last name available. What type of soil data was considered in the models given the large grids? So they imagine that the temperature would be more uniform than variation in soil properties within the grids.
Mhm. Um, I can't remember the specific ones. There are about 15 different variables that are like the most widely used on soil grids. So I pulled out all the variables that are related to soil, carbon, nitrogen content, like soil, clay, all those main variables. I'm happy to give a list of those if people are interested. But yeah, I can't remember them off the top of my head. It was all just the main ones in soil grid. Yeah.
Okay. And I can now clarify, uh, Bernard has been typing out the questions that are coming through in the background. That was from Omar Gutierrez del Arroyo.
Ah.
So we have a full name. So
Nice.
thanks, Omar.
Thank you.
And I'm gonna close then with a final question from Von Robinson, who asks, so these four categories you've identified, how do they relate to existing classifications based on growth strategies and associated, especially with water use efficiency among the various biomes? Can you say anything on that?
How do they relate to known variation in demographic strategies?
Yeah, so I guess there's two things wrapped up in there. Maybe how do these four categories relate to what's the known growth strategies, kind of
Mhm.
from theory, and is there any link that here between as related to water use efficiency and how that varies across the biomes? You can say
Mhm.
anything on that.
I mean, I guess loosely, I think it mirrors really intuitively what we would expect if you think about the structural complexity of boreal systems versus tropics, as far as more variation in the kind of maximal sizes of trees and the profile of forest communities. There's also, it mirrors, I think, quite well the longevity known from core data, so tree ring data, across gradients. So longer living species in the boreal and the tropic, or in the boreal and the temperate zone versus the tropics. And then all the really cool tree demography work from the tropics that I would say it really nicely matches the categorizations of what we see in the tropics, at least, of the really kind of a continuum, but more complexity beyond the fast slow. So there is a trade-off, but that there's multiple ways of maintaining high fitness that move beyond that simple continuum that we keep testing, but also showing that a lot of evidence for diversification. How that relates to water availability, I mean, I think we see that across the gradient as far as what we'd expect from hydraulics and physiology across the gradient of but we aren't, but I haven't tested the morphologic physiologic traits associated with water availability specifically. And again, I really think that would be a cool thing to do, incorporating those traits, because you would expect, particularly the tree height and the longevity, to be really associated with these physiologic traits, and that should be an interaction with environment. So it's not simply these traits, but the traits can vary depending on the context of the environment. So it should be an environmental by trait interaction.
Awesome. Thank you, Lalasia. And thank you very much for a really inspiring and interesting presentation and discussion. Really, really appreciate it. Glad you could come and present today.